Okay, now I'm going to welcome everyone to MSI's Thursday seminar series and remind you that we have monthly seminars lined up throughout the fall, every second Thursday of the month. And everyone invited this fall has been nominated by a member of the MSI community, which we're really excited about. And so we thought it only right that the person who nominated a given speaker be allowed to introduce them and give us their unique perspective on the guest we'll be presenting today. And so without further ado for today, I'll invite Heather to give the introduction for today's speaker.
Okay, can you hear me? All right, so it is my great pleasure today to introduce my friend and colleague, Dr. Dipti Nayak. Dr. Nayak is an assistant professor in the Division of Genetics, Genomics, and Development of Stanford's MCB department. She started that position in July of 2019, I believe. Before that, she was a Life Science Research Foundation Fellow, courtesy of the Simmons Foundation, and a Carl R. Woese Institute for Genomic Biology Fellow at the University of Illinois, working with Professor William Metcalf. She earned her PhD from the Department of Organismic and Evolutionary Biology, OEB, at Harvard, under the direction of Professor Chris Marks in 2014. As a grad student, she earned multiple awards for conference presentations and for her teaching. Just a little side note, we were grad students together in OEB, so we have many fond MSI memories of those days. And if I remember correctly, I think Dr. Nayak actually organized one of the journal clubs for MSI. Dr. Nayak also earned a master's degree from the Department of Civil and Environmental Engineering at Stanford in 2009. Her PhD research focused on using experimental evolution as a forward genetic screen to study metabolism and stress response in methylotrophs. Her postdoctoral research focused on the physiology and evolution of methanogenic archaea, employing a Cas9-based genome editing tools. And this was actually the first successful application of Cas9-mediated genome editing in a member of the archaeal domain. So that's pretty exciting. And we're going to hear about that, I think, today. In addition to publications in journals like ISME and PNAS, Dr. Nayak holds at least one patent related to her research. So Dr. Nayak studies methanogenic and methanotrophic archaea. She describes her work as being at the intersection of physiology and evolution, trying to understand the fundamental and distinctive biology of these organisms, which is key to designing optimized solutions for managing the effects of methane emissions. And on a more personal note, she has one of the sharpest minds I have ever encountered. She's a highly creative scientist, a talented teacher, excuse me. I know this firsthand as I relied heavily on her in more than one grad school course. And she is also a dedicated and compassionate mentor. So welcome, Dr. Nayak, and thank you so much for speaking to us today.
Thank you so much, Heather, for that very generous introduction. I think I'm so thrilled to be here. Let's see. Here we go. Okay. Can all of you see my screen? If someone can just say yes, that would be great.
Yes, I can see your screen.
Okay, perfect. Let's use the laser pointer. Okay, as Heather said, both of us were grad students in OEB at Harvard at the same time. And I definitely consider myself a product of the MSI. I think most of my microbiology exposure has come in some way, shape, or form through the MSI. And I'm like eternally indebted to many members of the MSI for kind of giving me those opportunities. And so with that, I'd like to start my talk today. Just want to make one quick correction. I'm an assistant professor in the Department of Molecular and Cell Biology at Berkeley, which is across the way from Stanford. And so with that, what I'd like to do is, take you back in time a little bit to introduce you to these unusual organisms that we study in my lab. And I want to take you back to the late 19th century, to the town of Manchester in England specifically. So at this point in time, the town sanctioned a famous painter by the name of Ford Madox Brown to paint 12 murals in the town hall that would depict the rich history of that area. The one that I'm showing you is the last of those 12. And in this one, you can see the very famous chemist, John Dalton, who came up with the atomic theory, performing a quintessential kind of microbiology experiment that many of us might know as the Volta experiment.
Dipti, I'm so sorry to interrupt. I just got a text asking me to ask you to speak up, if that's possible.
Okay.
Sincere apologies for that misspeaking.
No. Can we check the volume now? Then, I mean, I'm happy to go.
Yeah,
do a little.
I'm going to mute myself and let the MSI folks figure that one out, because I think I'm only hearing you through Zoom, not YouTube.
Okay.
Yeah, let's see. I can hear you okay, but I definitely want everybody else to be able to hear us as well. It looks like you're speaking straight into the computer. Is there any chance you have a set of headphones with a mic on them?
So I am wearing my headphones with a mic.
Oh, perfect. I couldn't see. Okay.
Yeah.
Pete says that's better, so I think you're all set.
Okay, great.
Great.
Cool. Yeah, and if anyone has, I guess I don't know how this works, but if anyone does have any issues, just feel free to chime in. I'm happy to be interrupted through this process.
Awesome. Thanks, Dipti.
Yeah. So, right. Going back, what this painting here shows is the chemist John Dalton, who came up with the atomic theory, performing what we call the Volta experiment. He's disturbing the sediment at the bottom of this marsh, and a gas would come bubbling out that you see this young person collect. This gas had interesting properties that were fascinating to John Dalton, in that it was extremely light and flammable, but that also made for an interesting visual spectacle. And that's kind of what fascinated maybe the non-scientists in the audience, and maybe was the reason why this whole kind of scientific experiment got memorialized in such a way. At this point in time, they didn't know what this gas was and kind of naively called it marsh fire gas, but is what we call methane today. And what fascinates me specifically about this piece of art is that at this point in time, they had no idea where this methane was coming from or how it was being produced. But there was something about this process that was kind of really fascinating. And it would take at least another century for people to figure out the source of this mystery methane. And it turns out that there are microorganisms that live in these marsh sediments and many other anoxic environments that produce methane gas as a byproduct of their metabolism. And here I'm just showing you some micrographs of what these microorganisms look like. And some of these initial studies that identified these microbes thought that they were bacteria because of their unicellular shape and the fact that they lacked any organelles, so that they were prokaryotic in their morphology. Turns out that that was also not the case. There was a surprise in store there, and in the discovery that was made, it was a discovery that was made together by Ralph Wolf and Carl Voss, in my opinion, because Ralph was the person who handed these methane-producing Archaea today to Carl Voss. And that kind of led to the tree of life as we know it. All of these methane-producing microorganisms that we've identified so far, all are members of this third domain of life, or the Archaea. So initially when the Archaea were discovered, so this is a tree from the late 1980s, all of these methane producers kind of branched on this one particular kind of clade of the Archaea within what we call the Archaeota. So what was kind of then considered was that the ability to produce methanes, this unusual metabolism, kind of evolved within these groups of Archaea, and that was about it. But in the last 30 years, so this is a tree of the Archaea from 2017, we've been able to populate the archaeal branch of the tree of life much more, specifically with the help of metagenomic sequencing. And now on this kind of diverse tree, when we map the genomic potential of organisms to perform methane metabolism, what we see is this trait is pretty diverse and broadly spread across this entire domain. And what I also do want to point out is that this trait is uniquely found in members of the archaeal domain. And what that really means, and one hypothesis that's kind of gaining favor amongst biologists now, is that it seems likely that the last common ancestor to all modern day Archaea potentially could have been a methanogen, or could have been performing some kind of methane metabolism. And what that means to me as someone who's interested in these groups of organisms is that the ability to produce methane or consume methane, as I will discuss later in my talk, And being an archaeon, the two kind of facets of these organisms are intricately connected. And when we study these organisms, we're not only just studying one aspect or the other, but we could be studying aspects of how this entire domain of life may have evolved in itself. The other thing that I want to point out about these methanogens is that while Dalton may have initially found them in these sediments at the bottom of a marsh, they're not just present in that environment. They're pretty much ubiquitous wherever we look. So if you look at anoxic kind of lakes in the Antarctic, anoxic Antarctic lakes, or hydrothermal vents at the bottom of the ocean, you'll isolate methanogens from them. In fact, when we think about it, the range, especially in terms of temperature and pH, that we see these methanogens kind of occupy on our planet would be one of the broadest known for any groups of organisms, including other diverse sets of microorganisms. In addition to being kind of widespread in the environment, the other interesting thing about these methanogens is they're often host associated. So you'll see methanogens associated with insects like termites, where they essentially are a part of the hindgut community and help these organisms metabolize complex substrates, as well as in cows that we often think of as a source of methane, but it's these methanogenic archaea that they have that produce the methane, not the cow itself. And finally, us, we have a substantial population of methanogens as a part of our own gut microbiota. And these methanogens in our gut are often associated with a healthy state, but also a disease state. There are certain studies that indicate that methanogens, the abundance of methanogens in our gut could be indicative of certain metabolic disorders like diabetes. But what I specifically want you to take away from this particular slide, is that living in all of these different environments, these methanogens release a lot of methane. And it's estimated that about a gigaton of methane is produced by these methanogens, which when you think of it, is about 80% of all the methane emitted on our planet. And methane is a really potent greenhouse gas. On a 10-year timeline, about 20 times more potent than CO2. And the reason I think today why we should be thinking about methane is these extremely concerning methane trends. So specifically in the last 5 years, the amount of methane in the atmosphere has been rising steeply. This particular graph here shows you kind of the recent trends, and the methane today in the atmosphere is at an all-time high. And the reason why that matters is when we think about some of the climate treaties, so when, things that we've heard of commonly in the news, like the Paris Agreement, to meet what we think is already a lofty goal, to limit the temperature rise by 1.5 degrees Celsius, the models that we're using assume that the methane emissions are actually going to decrease by a certain amount, 35% in one of the models, in the next few decades. But what's actually happening is just the polar opposite. So methane is a gas that I think we need to just pay more attention to. And if you're thinking about methane, we should really be thinking about these methanogens. So in my lab, what we're trying to do is kind of get a fundamental understanding of some of the processes in these methanogens that directly impact the net emissions of methane gas and then the contribution of methane gas to climate change. So today what I'm going to do for the rest of my talk is give you an overview of the techniques that we use in the lab and how we use them to study specific aspects of methanogenic metabolism. And towards the end, I want to spend maybe a minute or two in just kind of giving you an idea of where we're going with it right now. So with that, I want to talk to you about Cas9-based genome editing, as Heather mentioned. So based on my introduction, I think it's pretty evident that methanogens are an especially important group of microbes to be studying. One of the things that we as a collective group of microbiologists have been getting really good at is finding these methanogens in a range of different environments. And another thing that I think we've been getting really good at is isolating methanogens from the environment and sequencing their genomes. But what's become a bottleneck is that diversity that we can sample in terms of genomes, in terms of actually studying them and mapping genotype to phenotype, there's only two systems of methanogens that are genetically tractable that I've mentioned here, the Methanosarcina strains and the Methanococcus. So in my lab, what we do is we're taking these methanogen strains, which are abundant in nature and essentially encapsulate all the diversity of methanogenesis that we can see in terms of the different pathways that are available. And we're relying on genetic tools that were built by my postdoc advisor, Bill Metcalf, to essentially study these organisms. The one thing about the tools that existed earlier were that they were pretty laborious and time consuming. So if everything worked right the first time around, it was said that it would take about 3 to 4 months to make a mutant, but realistically maybe about half a year. And so when you think about grad students and postdoctoral trainees, it really limits kind of how far you can go with these organisms. So when I started as a postdoc in Bill's lab, CRISPR-Cas9 editing was just taking off. And we wanted to see if we could apply these techniques to methanogens and maybe speed up this process of making mutants so as to overcome this big bottleneck. Before I jump into the details of how we went about the process, I want to spend maybe a quick minute or two just giving you an overview of CRISPR-Cas editing. And so bear with me if you're extremely familiar with the process. So it involves two different components. It's extremely modular. So one of them is this protein called Cas9. And it's typically an open reading frame that's derived from a bacteria called Streptococcus pyogenes. The second component is a chimeric RNA molecule called single guide RNA. This molecule has a 20 base pair region that you can design to be homologous to any region of the chromosome that you want to edit, and an 80 base pair scaffold that binds to Cas9. So in this case that I've shown you here, when the guide is bound to the DNA, and also bound to the Cas9, it triggers a change in Cas9 and makes it behave like a pair of molecular scissors. And what happens then is that Cas9 essentially cuts the chromosome of the organism and generates what we call the double strand break, or a DSB. Now this double strand break linearizes the circular chromosome in bacteria and archaea, and that process is lethal to these group of organisms. So if you're just stuck here, the cells are essentially dead and cannot recover. But if to fix this break, you give the cells kind of a repair template with the mutation you want to introduce, the cells should be able to do so by a process that's known as homology dependent repair, or HDR. So using this process, we can essentially take a region of the chromosome and modify it to introduce the desired mutation. In eukaryotic organisms, there's another process called non-homologous end joining that I want to briefly mention, wherein you don't need a repair template. And this process essentially is a black box in that it kind of itself fixes this break by introducing small insertions or deletions. But this is something that we mostly don't have to worry about in bacteria and archaea, as this process is not present in these organisms. So with that brief overview, when we started working on this project, there were a couple of kind of outstanding questions that I also want to bring to your attention. The first one is that archaeal genomes are actually enriched in CRISPR systems, even more so than bacterial genomes. So what we wanted to know was whether the Cas9 homologs that belong to the type 2 system, which are notably absent in archaea, whether they'll even work in a methanogenic context, or is there some sort of an incompatibility that we should be concerned about? And the second thing we were concerned about was how little was known, or is still known, about how double strand break repair using homology dependent repair works in archaea. So the genes that are very well characterized in bacteria, like REC-A, for instance, don't exist in these organisms. So how this process operates and how efficient it is still kind of a black box. And so we were concerned that it might not work and not be compatible with some of these other processes. So I want to kind of walk you through how we went about this process by kind of showing you a proof of principle experiment that we did. The set of experiments that I want to talk to you about are with this particular genetic locus, which encodes an ABC transporter. The first thing that we did was designed a guide RNA to target this one gene within this 3-gene locus. And what we did was we introduced Cas9 and this guide into Methanogen acetovorans, our model organism, and asked whether the two together would generate a lethal double strand break. And on the next slide here, I'm showing you the data that we got back. the first thing, and I first want to orient you to how to interpret these data. So on the x-axis, what I'm showing you is the composition of a plasmid that we're introducing in the cells. So if it says plus, that means that that component of the CRISPR editing system is present on the plasmid. And on the y-axis, what I'm showing you is how many transformants we get back that have the plasmid based on a drug resistance marker that we use to select for the plasmid. So when we essentially introduce into cells a plasmid that contains Cas9, we get back about 1 million cells with the plasmid. So what that indicated to us is that Cas9 itself is not toxic to methanogens. So it's not that this protein is in some way, shape, or form incompatible in an archaeal context, which was a great start. The next thing to actually see whether this system works is we introduced Cas9 and the guide RNA together in these cells and performed the same kinds of transformations as we did here. But instead of 1 million cells, we got less than 10 cells every time we did this experiment. And the 4 order of magnitude drop in transformation efficiency here essentially told us that this plasmid is lethal to the cells. And so that means that this whole system is working. And it also told us that there is no cryptic non-homologous end joining that we know of or can detect of in these archaea, which was good to know because, again, as I said, DNA repair mechanisms in archaea are not well characterized. So we kind of ticked off this first box in these major questions that we were asking. And now there was a second question. So we know Cas9 works. We know that it can create a lethal double strand break. But what about the host itself? Can Methanogenic repair its DNA well? And can it recognize this break that was created by Cas9 and repair it with the template that we want to give it so that we can insert mutations? And so in orange here, I'm showing you a repair template that we provided. And we asked whether this particular template, which has a frame shift mutation and a diagnostic restriction site, whether the cells would be able to switch out the native locus for this modified locus. And so here are the data. So on this particular panel, I'm showing you a plasmid that has all of these components. And what you see on the y-axis is when this particular plasmid is introduced in the cells, the transformation efficiency goes up by about 4 orders of magnitude, indicating that the cells kind of take up this plasmid much more efficiently. And the reason for that is that the DNA that's damaged here can be fixed by repair with the template that we're providing. I'm going to cut a very long story short. What we essentially found was that all of the transformants that we screened here had the mutation. So we're working with a near 100% efficiency for CRISPR editing. At this point, since these proof of principle experiments, we've designed tens, if not hundreds of different mutants, and it works just as well for every locus that we've tested. We've sequenced the chromosome of multiple different organisms that have been genome edited. And unlike eukaryotic systems, we've not been able to identify any off-target effects. So not only is the system extremely efficient, it's also very precise. And the last thing, and maybe the most important thing that I want to highlight here, is that the reason why we went about this whole process was to increase the efficiency of genome editing in Methanosarcina. And I think this process is way more efficient. And it takes about 3 weeks versus 3 or 4 months now for us to make a mutant. And that's been really helpful, and it's been great because now we can do more in less time. I also want to leave you with one final thing about CRISPR editing. Since this process works so efficiently, we wanted to see if we can get a little more greedy with it. So far, what I've told you is what happens when we express one guide RNA and provide the template for that. But we wondered what could happen if you express multiple guide RNAs at once. So we kind of designed a set of different plasmids, which are expressing, in this case, 2 different guide RNAs. One set where each guide had its own promoter. So these arrows are showing promoters and its own terminator. So each guide would be a separate RNA molecule. We also expressed a version of the different guides where they're essentially arranged in an operonic fashion, where there's one promoter driving the expression of multiple guides. And we tested whether the cells would recognize different guides provided in these different forms. And the answer was yes. So we decided to go with this format for expressing multiple guides in the cell, just because from a molecular biology and cloning perspective, this is just way easier to construct. And so with this kind of framework, what I want to show you again is just a toy example of how we've been able to express multiple guides. So the genes here are not important. What I want you to pay attention to is how many guides were being expressed and how many transformants we get. So even the number of transformants that we get when we express 4 guides to delete 4 genes is the same as we get for 2 guides for deleting 2 genes. And we've been able to scale this up, and we've been able to use 5 to 6 guides to delete a few genes, introduce tags, and do all of that at once. And the power of this technique, I think, really lies in this maybe subtle phenomenon, is that traditionally in genetics, making mutants is a sequential process. You make a mutant and then you make another mutant. What we've been able to do with CRISPR is that we've been able to kind of make those processes happen in parallel. So that, if you want to access this really complex genotype that is multiple mutational steps away from the strain that you're working with, you can get to that now in weeks versus years that it would have taken earlier. So what that means from the viewpoint of someone who's interested in physiology is that you've kind of made more genetic space available and accessible for physiological studies. And so with that, I hope I've convinced you to some degree that we have this really nice CRISPR editing system that's up and running to study these methanocercina strains. And to the best of my knowledge, this is the only successful application of CRISPR-Cas editing in a member of the Archaea domain. And we're kind of building on this idea right now of multiplexed editing and hoping we can kind of start doing some genome-wide analyses to start figuring out what many of these genes of unknown function in these archaeal genomes actually do. So with that, towards the second half of my talk now, what I want to kind of talk about is how we've been able to apply these new CRISPR-Cas tools to kind of gain insights into the physiology of these methanogenic archaea. And the specific aspect of this process that I want to focus on is, again, the metabolic process by which these organisms make methane. So here, what I'm showing you is a very simplified cartoon of the diversity of methanogens in nature in terms of how they make methane. So there are some organisms out there that can take CO2 and reduce it in this stepwise fashion to make methane. There are others that can take methylated compounds and transfer the methyl group to form methane, either with an electron donor or by disproportionating it. And there are some methanogens that can take acetate and disproportionate it to make methane and CO2. I don't want to get into the mechanistic details of this process, but what I hope to kind of show you through this slide here is that all of these organisms first are obligate methanogens. So far, we have not been able to isolate a methanogen or find one based on genomic potential that can do more than just methanogenesis. So the ability to produce methane is kind of intricately coupled in these organisms to the production of a sodium or a proton motive force, which is required to produce ATP, as well as the production of biomass. So if these organisms don't make methane anymore, they're not going to be able to survive or reproduce. And despite the fact that there are multiple different pathways in different organisms to produce methane, there's one step of this process, independent of which organism you're looking at or which process you're looking at, that is universally conserved, which is this last step, which is the production of methane itself. So the step in the cell that catalyzes the production of methane is catalyzed by this enzyme called methyl coenzyme M reductase, or MCR. MCR to me is an absolutely fascinating enzyme. And in terms of its importance, I think it kind of ranks right up there with Rubisco. So this enzyme, as I mentioned before, is present in all methanogens. And if you think about the fact that 80% of the methane on our planet is coming from methanogens, if you think about chemically, 80% of that methane is being produced by this one enzyme, MCR. So this on the left here, what I'm showing you is the structure of MCR itself. So each molecule of MCR contains 3 subunits that I've highlighted in different colors. And each molecule has 2 of these 3 subunits. So it's a dimer or trimers, as we would like to call it. Now, when you think about how this enzyme makes methane, one of the 3 subunits becomes more important, which is the alpha subunit in green here, because that's where the active site of the enzyme resides. So on the next slide, what I'm going to do is show you what the active site looks like. And what I want you to envision is that you're taking away the beta and the gamma subunits and looking at the enzyme top down. And this is what the enzyme would look like. So what I've highlighted in these different colors are the substrates for the enzyme. And in addition to that, you see this molecule that's non-covalently bound called factor F430. So F430 looks a lot like some of these other molecules in nature that we might be familiar with, like heme and chlorophyll, in that it's a tetrapyrrole. So it has 4 of these 5 carbon chains in the molecule. But unlike heme, F430 coordinates a transition metal called nickel. And this might be a subtle change, but it actually has kind of dramatic implications in terms of thinking about MCR. So one thing that scientists who've been studying MCR have identified is that this particular nickel is absolutely critical for this enzyme to work. And not just that, the redox state of this nickel is important too. So this nickel has to be in its +1 oxidation state for the enzyme to be active. But when you think about the redox chemistry of nickel, that becomes important here. This particular redox couple, specifically nickel 2, nickel 1, has a very low redox potential. And what that means that this nickel is very prone to oxidative inactivation. So the cells of methanogens have figured out a way to keep that nickel reduced. But we as scientists, when we break open these cells and try to study this enzyme in vitro, then this nickel gets oxidized. And it's taken researchers, decades to figure out how to keep this nickel reduced outside of the cell. And even though there are solutions that we now have found, it's still extremely cumbersome to study this enzyme because of this nickel and its ability to get oxidized, and then the enzyme falls apart as a result. As a result, many aspects of this enzyme are still poorly understood. And there's been some recent studies that have highlighted a possible mechanism for methane formation, which involves the formation of a methyl radical as an intermediate. One aspect of this enzyme that kind of drew my attention was that in the active site, as I'm going to highlight further, many of the amino acids kind of undergo post-translational modifications, and they are likely, and they were thought to be very important in how this enzyme works, and specifically in certain steps of catalysis. So here what I've done is taken the same figure as before, but just kind of highlighted the amino acid residues that undergo post-translational modifications in the MCR derived from Methanosarcina Archaea. 4 of these 5 modifications that are common in nature in different MCRs are methylations. I've just highlighted them here. And what I want to point out, because I'm not going to get into the details of these methylations today, is that none of these methylations are universally conserved in all MCRs that have been studied. There's often some combination of these 4 that are found, but typically not all 4 are found altogether. So for instance, in Archaea, we find this methylhistidine, we find the methylcysteine and the methylarginine, but the methylglutamine is absent. There's a 5th mod, so altogether, these methylations are present, but not universally conserved. There's a 5th modification that I want to discuss today, which is this thioglycine, which unlike these methylations is universally conserved in all of the different MCRs that span kind of the spectrum of diversity that we see in nature. And this thioglycine, if you think about it, is a very simple modification. So it's the conversion of a C double bond O in a thioamide bond to a C double bond S to give you a thioamide bond. So it's essentially a switch from an oxygen to a sulfur. What's particularly unusual about this thioglycine is that there's no other known protein that we know of so far that has this modification. So this modification is especially unique to MCR. And most importantly, because it is so unique and unusual, and kind of where it's sitting in that little channel that leads to the formation of methane, for a long time, it was speculated that this sulfur somehow has a role to play in the electron relay that leads to eventually the reduction of a methyl to form methane. But it wasn't quite sure how that would work. And so our approach to this was to essentially figure out the source of that glycine, the thioglycine. And in collaboration with Doug Mitchell's lab, who studies natural products, one thing we observed was that there's this molecule called thioviridamide that's produced by a group of streptomyces. So this is a natural product that's produced by these streptomyces called Streptomyces olivoviridis that have the same chemistry that we observe in MCR. So that the presence of this C double bond S or this thioamide bond. And what some researchers in this paper that I've highlighted here had found was this gene cluster from A to O in this particular organism, Streptomyces olivoviridis, that is responsible for making this product that I'm showing you here. These researchers had even gone as far as to indicate that there were two genes within this cluster, TVA, H, and I, so these two genes right here, that had strong homology to proteins that were found in methanogens. And so this was not a coincidence. I just want to remind you, Streptomyces and methanogens are very distantly related organisms. The only thing that we could think of that these two organisms would share homology for is the ability to perform this chemistry. So we thought that these two genes are likely candidates in methanogens for making that modification in MCR. And so I'm going to refer to these genes as YCAO and TFUA. So using CRISPR, we essentially first identified this genetic locus in our model organism, Methanosarsina acetoborans. And using CRISPR, we designed constructs to delete it. And maybe the first surprising thing that we found was that we could delete these two genes. So it looks like these two genes are not essential in the cell. And so that was surprising because we thought that this thioglycine was essential for MCR to work. MCR is essential for the cell to survive. So does that mean that thioglycine is not important? But kind of the obvious question is maybe, why CAO and TFU are not doing the thing that we think it does. So we had to go through a pretty elaborate process to figure out what these two genes do. So again, going back to MCR, what we did was figure out a way to tag these genes, again, using CRISPR. And with that, what we could do here is take the cell lysate and use this tag that we've introduced in these genes to purify MCR. And this was great. We could purify MCR with this tag now in 30 minutes, where before it would take researchers 24 hours to do it with ammonium sulfate. We then took the alpha subunit of MCR, so this particular little protein here, we digested that protein and specifically looked for the region of the protein that was supposed to contain thioglycine. And that particular region, that particular peptide of interest, if we wanted to look at what its molecular weight would be. So if it has thioglycine in blue, it would have a molecular weight of 34-32 Daltons. And if it did not have thioglycine, if it just has regular glycine, the molecular weight would drop by 16 Daltons. And so using MALDI-TOF, what I'm showing you here is what the molecular weight of that peptide is. So in the wild type cells, you see the weight of the peptide as 34-32, which means that it actually has thioglycine, which we've known of. But in each of these mutants that either lack both of these genes, YCAO and TFUA, or each of these individually, we only see the peptide that corresponds to just regular glycine. And so what this told us is that indeed, by knocking out these two genes, you get rid of that thioamide modification in glycine, and MCR just has a regular old glycine in its place. So you might think that's a simple gene type to phenotype, but I do want to take you back now and say that, what we've shown here is something really important. What we've shown is that for the longest time, people thought that this thioglycine was important, that it was catalyzing this electron relay. And we've said that, no, that's not the case. An MCR with the glycine is just as good as an MCR without the thioglycine. And, all of those hypotheses are actually incorrect. So the question then is, what does thioglycine do? Why does MCR bother going through this process? So initially, we were maybe a little concerned that we'd run into what are called evolutionary spandrels. So something that's, bright and shiny and unique, but may not have a particularly important functional role. But then as we dug deeper, I'm going to leave you with this one final kind of chart that we think tells us what this thioglycine might be doing. So here, what I'm showing you are the growth rates for cells that are growing on the same substrate, but at different temperatures from 29 degrees to 45. The optimal growth temperature for these organisms is 36 or 37 degrees Celsius. And what you see specifically for the mutant that lacks this thioglycine modification, so the mutant in green here, is that as the temperature increases, these cells are not able to grow as well as the wild type. So they do have this temperature specific growth defect. And so the hypothesis that we're now entertaining for thioglycine is that it might have a role in maintaining the thermal stability of this enzyme and help it work under certain conditions, specifically maybe high temperatures. And it's not required in catalysis as it was previously believed. So with that, I'd like to kind of maybe summarize this part of my talk and say that using CRISPR, we've been able to kind of identify and genetically manipulate genes that are involved in controlling the activity and the conformational stability of MCR. And this CRISPR-Cas editing is giving us a window to understand this extremely important enzyme. So in the last few minutes, what I would like to do is quickly maybe go through kind of where my lab is right now and what we're working on. And to introduce some of an ongoing project in my lab, I have to introduce another set of organisms. Heather mentioned these organisms right at the beginning when she was introducing me. And I want to bring this idea that, you know, methane metabolism is prevalent in the Archaea. And while a lot of our focus is on these Archaea that produce methane, the methanogens, there's another group called the ANME, which are anaerobic methane oxidizing Archaea, that are very closely related to our methanogens. But as the name suggests, these organisms essentially do the opposite of what methanogens do. So they don't produce methane, they actually consume methane. And unlike our methanogens, they're much slower growing. And so we haven't been able to isolate any pure cultures of these ANME yet. And what that means in terms of, again, thinking about genotype to phenotype mapping, very many of the mechanistic details of how these ANME consume methane are still poorly understood. What we know so far about these ANME, based on kind of their genomic composition, is that they look a lot like methanogens. Again, I do not want you to kind of get confused maybe by all the symbols here. What I would hope to kind of indicate here is that if you look at the genome of a methanogen, and if you see all the genes here that are conserved and are used for the reduction of CO2 to methane, the same genes are present in these ANME Archaea that are methane consuming, and they just operate in reverse to take methane and oxidize it to CO2. And so what that means is that in principle, based on their genomic content, these ANME essentially perform what we call reverse methanogenesis. But the process is a little more complicated because if you think about methanogenesis and everything we know about it, it's an energetically favorable process. But these ANME Archaea, what they have to do is take all of those same genes and run them in reverse. So thermodynamically, they're essentially, if everything was operating in reverse, the whole process should be energetically unfavorable. So there must be new and interesting innovations that ANME are making, and we know some of them already, that help them kind of overcome some of these thermodynamic hurdles. What we're hoping to do in my lab, without getting into too many details, is find what some of these key innovations might be, introduce them in a methanosarcina strain, which is genetically tractable, that as I've discussed in the last 30 minutes or so, and eventually maybe work towards this lofty goal of engineering this methanosarcina to become an ANME de novo in the lab, so that we can really understand how this metabolic transition happens in nature. And so with that, I'd like to thank many of my collaborators and mentors who I've had the tremendous fortune to work with, which includes Bill, my amazing postdoc mentor, Doug Mitchell and Satish Nayak at Illinois, who kind of helped us a lot with the MCR post-translational modification project that is still ongoing in Bill's lab. Postdocs with Doug and Satish, as well as Victoria Orphan, with whom we're collaborating on this ANME project that I mentioned recently, and Adam Arkin's group, with whom we're working kind of to develop genome-wide functional screens for methanosarcina. I'd also like to thank members of my new-ish lab and all of these amazing funding sources through my postdoc and now as an assistant professor that have enabled us to kind of do all these fun scientific experiments that keep us happy and going, especially in these crazy times. And so with that, I'd like to thank you all for your attention, and I'm happy to take any questions if you might have them.
That was a really stellar talk. Thank you so much. I'll remind and invite and encourage everyone who is watching on YouTube that they can submit questions through the chat, and we can start with one that came in during your talk, which is someone asking, what do you think about the application of such CRISPR-Cas tools for other methanogens such as those in the gut?
That's a great question. Yes, so I think so right now what we know about methanogens in the gut is that they're not genetically, no one has been able to genetically modify them yet. There are certain barriers to that process. One of them is that many of these gut-associated methanogens have a very thick pseudopeptidoglycan layer, and it's hard to get DNA across. So I think before CRISPR-Cas editing, people might have to kind of think about how to deliver DNA to these organisms reliably. And once that process is worked out, I think the next step would be to try CRISPR-Cas editing.
That makes sense. Thank you. I had a question also. I'm really curious about if you know what factors contribute to the increased precision of CRISPR-Cas gene editing in Archaea relative to in other cell types.
Yeah, that's a great question. This is just a speculation. I think it has something to do with how they repair DNA. There's a few papers out there. There's certain aspects of the DNA repair mechanism that resemble eukaryotic genes, but there are certain parts that are uniquely archaeal that have been characterized in certain other archaea that, like HER-A and NER-A, that are not present in acetoborans. And so there might be certain, and other methanocyclino strains. So there might be certain counterparts that we haven't been able to identify yet because no one's looked at it, that are important in this process and that could be guiding the precision. Yeah, it's not been looked into. I think it's fascinating. And it was surprising to us. I would also add that because we were pretty sure that there'd be some off-target activity and we just haven't been able to find it.
That's a really good surprise.
Yes, it's one of the better ones.
We got a question here. I also work with a protein in anaerobic bacteria. I'm wondering how to purify MCR under strict anaerobic conditions.
Yeah, that's a great question. I wish I had, I can go on forever about this. So the thing about MCR is that there's that nickel that gets oxidized and you have to keep it reduced. Traditional reductants that we think of, like cysteine, DTT, they don't work with MCR. So one of the biggest hurdles there was to find a reductant that would work. The one thing that people have shown works efficiently is this chemical called titanium chloride. And it only works at certain pH, at an alkaline pH, so greater than 7.5. And it works for some MCRs, but not the other. So that's what I can tell about MCR without getting into the gory details of the process. And I think sometimes that's what it takes. It's trying to find something that's a good chemical proxy for the cellular environment, and that can be challenging.
Mm-hmm. Definitely. That's actually related to the next question from Roxanne, which is how do methanogens protect their MCR from oxidation? Do you think conformational stability mediated by thioglycine is also important for oxygen exposure?
That's a great question. Yeah, so there's a paper that came out recently that shows that one of these other modifications, this methyl arginine modification that is found in these methanogenic archaea, but seems to be absent from these ANMI MCRs. That one, in that paper, what they showed was when they exposed the cells that have that modification to hydrogen peroxide in the medium versus cells that don't have that modifications to hydrogen peroxide, those that had that methyl arginine could survive peroxide stress a little better. So that does seem to indicate that maybe some of these modifications have a role in mediating oxidative stress response, specifically with respect to MCR. So that's what I can say based on literature. Broadly speaking, I think the question that I find interesting there is, how does the cell do this crazy chemical feat? And there must be specific reductants that the cell uses. And what I didn't mention maybe clearly in my talk was that this nickel is buried deep within the enzyme, so it's not easy to access. So how does that reductant access that nickel and keep it reduced? Those are all interesting questions in the field. We think that there's specific proteins involved in the process. It has something to do with how MCR folds and things like that, but it's still, the work is still all very preliminary for me to kind of make any definitive claims.
Mm-hmm. Awesome. Duncan is asking, do you think that the slow growth rate of the ANMI will be a problem, practically speaking, for the genetic manipulation?
Yes, I definitely think that's a problem. But I think what we're maybe more interested in is not, at least as a first pass, not really getting growth, but just making sure that electrons move in the reverse direction. So as long as we can get stable conversion of, methane to CO2 in resting cell suspensions without forcing cells to grow, I think we can override this issue of slow growth in the lab.
Nice, that makes sense. I was curious if you could talk a little bit more about, I was not familiar with the idea of the last common ancestor being a methane metabolizer. So I was just curious if we could talk a little more about that idea, or where are people actively doing research in that area, or where those ideas are coming from?
Yeah, so definitely not something I'm working on. So this is kind of my reading of the literature. I think from what I can read, and if there's someone watching who would like to correct me, please do. I think it's just kind of, as we're sampling more and more of the diversity out there, and as we're learning more about just the diversity of MCR and diverse groups of organisms, what was thought initially, methanogenesis is a very complex trait. It requires all of these specialized enzymes that have their unique cofactors. So you need all the genes for the biosynthesis of these cofactors. So it was thought that it showed up once in this one particular group of Archaea, and that was about it. But now it looks like many diverse groups of methanogen-like organisms are there in nature. We don't know if they do methanogenesis, but something similar with a similar group of enzymes. And it looks like they're often separated by groups of organisms that don't have this trait. So by looking at that, one can make this observation that if this trait is so hard to have been horizontally transferred over and over again, it's likely that what may have happened is that this trait was lost in those organisms that don't have it. And as the diversity of this particular metabolism is becoming apparent, we're kind of entertaining this idea that the last common ancestor who shared this trait was way further back in time than we may have initially believed. And that's kind of leading to this hypothesis that the certain ancestral nodes within the Archaea that we initially thought might not be methanogenic, might be.
Awesome. So I always also like to ask, this is a more general question, but sometimes when I give a talk, especially one with, say a 30-minute time frame or on the shorter side, there are always things that you can't fit in. And is there something you feel especially excited to talk about in the last few minutes that you wish you could have fit in the talk, or something, or an experiment, or a direction that you all are taking? You talked some about that at the end that you'd want to expand on a little bit.
Okay, how many minutes do I have for this?
4.
Okay, well, 4 minutes is hard, but yeah, I think something that I'm particularly interested in is, myself included, we've really kind of honed in on methanogenesis as a biochemical pathway that operates in these organisms, and specifically looking at how these genes work. I think maybe one layer of the process that I think we haven't really been thinking of is all of these genes operate in the context of an archaeal kind of an archaeal cell. And what that means is that certain processes that are core housekeeping, such as gene regulation or double-strand break repair, as I mentioned in the context of CRISPR, happen very differently in the cell than, say, a bacterial cell. And we haven't been kind of working at that intersection of, say, how do genes involved in methanogenesis get regulated? And the reason why that process is important and interesting is, these genes are kind of arranged in this operonic fashion, often like bacteria. But DNA transcription happens with an RNA polymerase that's not at all like bacteria. So like these archaea don't have sigma factors that we're so used to thinking of. So what is the process by which they mediate, gene regulation? And if we kind of understand that, we won't just kind of tie that into methanogenesis, but maybe tie it back further into just how these processes work in an archaeal context, which is often kind of a rate limiting step when we start kind of, looking at genes and finding genes of interest, is what does this gene do and how does it work?
Completely. Awesome. Well, this has been really fantastic. Thank you so much for joining us. And I'm really grateful to Heather for having nominated you for our fall series here. I'll just remind everyone that we have a continuing awesome lineup of speakers every second Thursday of the month throughout the fall term. And so we'll look forward to having you all join us each month for a new seminar on more cutting edge microbial science being done across the country. So thanks so much, Dipti, and have a great afternoon.
Thank you.